So good morning, students. Today we are going to discuss about molecular basis of inheritance, chapter 6. And here in this chapter, we are going to deal with factor, gene, DNA, chromatin reticulum, euchromatin and heterochromatin, nucleosome and histone protein, macromolecules of DNA, nucleoside and nucleotides, DNA, polynucleotide chain and DNA structure. So in our next slide, as you see in our next slide, we are going to show the part of DNA that actually involved in the formation of chromosomes. So in normal nucleus, within the nucleus, we found it is the structure shown in electron microscopy. The chromosomes are not distinct. Here, many elongated thread-like structure found inside the nucleus. These are called chromatin reticulum. And if we see under high magnification of electron microscope, then each chromatin reticulum consists of a long beaded chain-like structure. And this beaded chain-like structure are always linked by linker DNA with one another, and these beads are called nucleosome. So nucleosomes are present in chromatin reticulum or chromatin fiber, and in the next slide here you see the structure of nucleosome. Actually, DNA form wrapped around a histone molecular or pore. This is the histone octamer, and the strands of DNA is linked to one another by H1 histone protein. So here is the structure of the bead of nucleosome. Here eight histone cores are found. They are found in 4 pairs or total 8 histones, H2A, H2B, H4 and H3. That means H2A, H2B, H3 and H4 are the 8 types of histone protein found in the DNA beads, and at the center or inner core is called a new body, and the DNA strands wrap around the nucleosome in 2 turns, and the DNA strands are linked by another short DNA strand, that is H1 consists of H1 histone protein, and it is called linker DNA. So this part is given in detail in the textbook of NCERT. There is also in concept of biology. You can go through it to find detail of their positive and negative part of the DNA structures. So how many bits or nucleosomes are present in a particular human cell? In a human cell, the nucleosomes or bits part actually 200 BP. BP means base pairs are present in one bit. So in case of human cell, we found 6.6 into 10 to the power 9 BP are present in. So its total will be 3.3 into 10 to the power 7 bits in a human cell. And theoretically, approximately, we can say that in per human cell, it has how many nucleosomes? 3.3 into 10 to the power 7 bits are present in a human cell. So next slide, here you see the nucleotides. Nucleotides are the macromolecules of DNA, and basically before the formation of nucleotide is from the nucleosides. Nucleosides, we know that when nitrogenous base linked with the deoxyribose, it is called nucleosides. There are 4 nucleosides found in DNA. Those are adenosine, guanosine, cytidine, and thymidine. And these nucleosides are converted into inactive form in the nucleus. Those are deoxyadenosine monophosphate, deoxyguanidine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. So nucleosides are 4 types in DNA, and they, when react with phosphoric acid, that converted into nucleotides. In presence of an enzyme phosphorylase, and energy is required for the conversion of monophosphate into triphosphate during the replication of DNA and during the formation of mRNA, etc. When they are inactive, they remain in the nucleus in form of AMP, GMP, CMP, and TMP. And D small letter denotes that the sugar is deoxyribose sugar. So when it converted into active form, or when the nucleosides are linked with phosphoric acid, it converted into nucleotides. 4 nucleotides are found in DNA. Those are D-ATP, D-GTP, D-CTP, and D-TTP. D-ATP is deoxyadenosine triphosphate, D-GTP, deoxyguanidine triphosphate, D-CTP, deoxycytidine triphosphate, and D-TTP, deoxythymidine triphosphate. So these 4 nucleotides are found in DNA. So since in this chapter we are discussing about the chromatin reticulum, so if we see the nucleus of a cell, that is eukaryotic cell, here we found 2 types of chromatin reticulum. One is euchromatin and another is heterochromatin. So we have to find out what is the difference between euchromatin and heterochromatin. So in our WhatsApp class, we are going to discuss in detail what are the difference between euchromatin and heterochromatin. One is genetically active, other is genetically inactive. In one case, DNA is more than histone protein. In another case, protein part is more than DNA. And so one is brightly colored, other is dully colored. So these differences we can get it from the textbook. So in the next slide, here the structure of the DNA. So if you ask or describe the structure of DNA, then you have to draw this diagram. First you see, since in DNA 4 nitrogenous bases are found. What are those 4 nitrogenous bases? A, G, T, and C. That means instead of writing as adenine, guanine, cytosine, thymine, we can write also the first letter of the nitrogenous base. So here we write A, G, T, and C. So during the formation of nucleoside, the adenosine nitrogenous base linked with the pentose sugar. This is the structure of pentose sugar. That means 5 carbon sugar, and it is sugar is deoxyribose sugar. The first position of the carbon linked to the nitrogenous base, and the link, there is a bond is formed that is called glycosidic bond. So when there is a union between nitrogenous base and deoxyribose sugars, it is called nucleoside. But when this nucleoside linked with phosphoric acid, then this whole structure from nitrogenous base, pentose sugar, and phosphoric acid, this structure is called nucleotide. So nucleotide is the macromolecule of DNA. In DNA, 2 strands are found. That means DNA is double-stranded. We always say it, and when it draw, the 2 strand we show in diagram. So here is the 1 strand, here is the 2nd strand. In the first strand, you see from I start from this point. This is the cytosine nitrogenous base linked to the pentose sugar by glycosidic bond, and this nucleoside linked to the phosphoric acid at what position? At 5th position of the carbon sugar. And this phosphoric acid, when linked to the nucleoside, it forms another bond that is called phosphodiester bond. And again, this phosphoric acid linked to the upper nucleoside by the help of phosphodiester bond, but at what point? But at 3rd position of the deoxyribose sugar. So phosphoric acid in one arm linked with the 3rd position of lower deoxyribose sugar, and at the upper nucleotide it linked to the 3rd position of the deoxyribose sugar. So the diagram shows this: the linkage of the backbone of the DNA of one strand is always 3 days 5 days. Here you see 3 days 5 days. Is it clear? And in the another strand also, these 2 strands are always anti-parallel. That means the strands are opposite. Here the upper strand is 3 days and lower strand is 5 days. That means at the same position, at first position, the nitrogenous base linked with the deoxyribose sugar, but here it linked with the 5th position and the 3rd position, 5th and 3rd, 5th and 3rd in this way. So these 2 strands are anti-parallel. So in double DNA, we get double strand, and these 2 double strand again linked with each other. by a hydrogen bond. That means the nitrogenous base always pair up in DNA. A always pair with T, that means adenosine paired with thymine, guanosine paired with cytosine, again thymine paired with adenosine, and cytosine paired with guanine. So here we write here A paired with T, and this bond is called a hydrogen bond. In case of A and T, there is a double hydrogen bond is found, and in case of C and G, triple hydrogen bond is found. That means A always complementary to T, and T always complementary to A. Similarly, G always complementary to C, and C always complementary to G. So this is the main structure of the DNA. Next here, see, although in the earlier diagram we draw the diagram in a straight manner, but actually DNA double strand structure always found in helix form, like a ladder, but in helical manner. So here you see the 4 nitrogenous bases are distinctly given in the diagram by a colorful manner, and here this one strand turns suddenly, and this how they turn? When there are 10 nucleotides in this gap, then it turns in right direction. Again left direction, again right direction, in this way. So in each turn, there is 10 nitrogenous bases, and the internucleotide distance between two, it is the 0.34. And the per turn, how many? 34 angstrom is found. And the diameter of the two strands is 20 angstrom. So this is basically very static, and if you tend to describe, asked to describe the structure of DNA, then you can say the first point, DNA is double-stranded structure. The pairing of the nitrogenous bases are always complementary. A always paired with double bond with T, and C always paired with triple bond, and that is hydrogen with G. And the two strands of DNA are antiparallel. One is 3 days 5 days strand, and another is 5 days 3 days strand. And what is the diameter of the two strand? 20 angstrom. And what is the length of the strand per turn? That is 34 angstrom. And what is the internucleotide distance between the two nucleotides? That is 0.34 angstrom or nanometer. So this is the basic structure. And what type of sugar is found in DNA? That is deoxyribose sugar. Why it is called deoxyribose sugar? Because at second position of the carbon, one oxygen is less, and its molecular formula is C5H10O4. And what are the nitrogenous bases found in DNA? That is A, T, C, and G. And what is the nucleosides found in DNA? Adenosine, guanosine, cytidine, and thymidine. And what are the nucleotides of DNA? DATP, DGTP, DCTP, and DTTP. So this is the class today we discussed, and I think you got well about this part. And in next class we are going to discuss about the replication of DNA.